Can we design plants to soak up pollution? Is a particular protein structure key to developing a vaccine for Lyme disease? How does sunburn lead to skin cancer? What are the biological effects of radiation in space? These are just some of the questions posed by scientists in the biology department at Brookhaven National Laboratory. With a distinguished history of discovery, the biology department specializes in understanding proteins and DNA, the basis of heredity. Researchers study molecular structure, genetics, DNA damage and repair, and the cellular biology of bacteria, plants, and animals. Each year, about 50 scientists from around the world and from Brookhaven Lab itself use the department's Scanning Transmission Electron Microscope, called STEM, to image biological specimens. Electron microscopes are like conventional optical microscopes, except that they use electrons instead of light to view a biological sample. A magnetic lens focuses the electron beam onto the sample, triggering interactions that are picked up by a detector and formed into an image. In STEM, the electron beam is scanned across the sample, and a sophisticated detection system records the individual electrons that interact with it, enabling scientists to see the intricate details that make up living organisms. Light microscopes are limited to 1,000 times magnification, not powerful enough to see the fine structures of cells. STEM can magnify samples 10 million times, mighty enough to see single atoms. That's something few microscopes in the world can do. Brookhaven biologist Joseph Wall, one of the inventors of STEM, explains why the microscope is important.
Welcome to the STEM facility. I'm Dr. Joe Wall. I've designed and built this microscope, which you see in the background. It's 100% custom made, as optimized for looking at biological specimens. It's unique. It's the only one in the world exactly like this. We've set it up to look at biological specimens from many different sources and worked out new structures. For instance, virus particles are put together according to certain rigid rules, and by measuring the size and the molecular weight of the virus particles with the STEM, we can say how they're organized internally.